Good evening. This is the 5th of February, 2026, and we are covering a paper for Journal Club in the Science Academy. That paper's title is difficult. C4D, a high affinity LILRB2 ligand is elevated in Alzheimer's disease and mediates synapse pruning. This is Brott et al. It was published. Let's see. It was published. It was accepted in August of 25, and it was published. It doesn't really say, does it? It was accepted August 20th, 2025, in the Proceedings of the National Academy of Science. This is the most prestigious group of scientists in the country. Ray Deshays, who was here 2 weeks ago, is a member of the proceeding of the National Academy of Science. He's also a member of the National Academy, which is a different thing. This was contributed by the corresponding author. So if you look at the last name on the paper, the Carla Schatz is the contributing author, and she's the corresponding author. She's also a member of the National Academy. Chris and Marcello, were you planning on talking about Carla's work at all?
No, we did not plan on it.
Okay, then why don't you go ahead and start sharing your screen? It's If you're ready for your thing, and I'll just chime in because she's pretty important.
Marcello, do you want to present?
Yeah. How do I screen share?
Off the chart.
There's a green button at the bottom. Just move your mouse
Oh,
over it. It says share.
it's working.
Yeah.
Now hit slideshow. Good.
Yeah.
Okay. Hi, everybody. My name is Christopher Forges, and my partner for this project, name is Marcello Ciriello. And today we will be presenting a paper about C4D, which is a ligand, and how it is elevated in Alzheimer's disease, and it mediates synapse pruning.
So the focus of this paper is the scientists are trying to determine whether or not C4D can bind directly to the LI-LRB2 and PIRB on neurons. And they were trying to answer the questions if it directly binds to these receptors, where does it bind on these receptors, and if it actually causes synapse loss. So a synapse is like a junction that connects two neurons, as you can see in the picture. And synapse pruning is a process by which your brain like removes the weak or unused synapses. So this is really important in Alzheimer's. It's a mental disorder where your brain like slowly deteriorates, just like the picture. And it usually starts to happen around the age of 65. The brain cells die out, which leads to long and short-term memory loss, as well as a lack in ability to complete everyday tasks since your brain's like controlling everything.
Okay, so the complement system is also a part of the body that is affected, or a system of the body that is affected by Alzheimer's disease. And the complement system is an immune-related system, which is important when it comes to protecting and development. And this is important for this article because something the system does is it helps remove extra synapses. And when it is under the influence of Alzheimer's disease, this system can become abnormally active, which can result in removal of too many synapses. So,
What?
oh, my bad. So some key molecules that you guys should know for this presentation are LILRB2, PIRB, C4D, C4, and PSD96. So LILR2 or LILRB2 is a receptor which can be found on human neurons. And then similar to this is PIRB, which is basically the same thing, but instead of human receptors, it is mice receptors, which is common, like a common substitute for humans, like for testing. Next up is the C4D, which is a ligand for LILRB2. And this is a fragment produced when the protein C4 is activated. And then finally is PSD95, which is a protein found at synapses and is used by scientists as a marker to measure synapse number and health. So now I'm going to talk about why these key molecules are important for the article. So LILRB2 is the missing link that connects the immune system to synapse loss. And then again, PIRB is similar, but instead of being human, it is the mouse version. Next is C4D. And then this is important because it is the immune signal that starts the entire pathway, which leads to synapse loss. And finally, PSD95, which again is how the authors end up proving that synapses are actually being lost. And then does anybody want to present figure 1? Andrew?
Right. So figure 1A is showing that a protein C4 is cut and rearranged to make C4B plus C4A, which is then turned to IC4B, and then which is turned to C4C, and the end product, the most important one, is C4D. It's a little blue part that's cut off from C4B, and then obviously the end product after the final reaction. So figure B takes the products in C4 itself and it puts onto a membrane, which is basically made of a certain material. It's fake. And then once they are put onto the membrane, you also have LR2B or LRB2 and SPIRB. So when the receptor sticks, the dark spots appear. Only C4D and um, LR2B and SPIRB stick around. Compared to the other ones, they do not. So that must mean these molecules have a, connection to each other. Now in 1C, they actually use a real membrane and more overlap in the colors means that there's a lot more, again, connection and binding. So as you can see for ORB2, there's a bunch of, red, blue, and, purple overlapping, which means that C4D clearly has strong connection to LRB2 compared to the other ones like LRB3 and LRB1.
Yeah, so if I can interrupt for a second, you guys can hear me, right? This is probably can you guys go back to the first slide? I should. Right, so if you can see that, let's see, draw. I want to draw. Good. Okay, can you see that right here? That's Carla Schatz. Carla Schatz is a legend. She has a really good chance of winning the Nobel Prize. Her stuff's amazing. So just knowing that, like I know her, I don't think I've ever met her personally, but I know who she is because I've read a lot of her stuff because I'm a neuroscientist. And she's very famous and has done a lot of great stuff. But she's the one who contributed this. If you see down below, it says contributed by Carl Schatz, right? She's gonna in the proceedings of the National Academy of Science, it was kind of meant, it was designed as a journal to really highlight the work of the National Academy. And so until as late as 2008, what they were doing this weird thing that no other journal was doing, which was if a member of the National Academy submitted something, it would not be reviewed. It would just be accepted. Now you can imagine that that's pretty shady, because you would want science is actually helped by doing a review and receiving criticism of your work. And this is a way of just kind of skipping that. But since 2008, you can't do that. It still needs to be reviewed. So you can see that she contributed it, but it was reviewed by two other kind of famous guys, Jonathan Kipnis and Aaron Schumann. And so, sorry, I thought I heard my baby cry. So a couple things that you can go into this thinking. One, positively, if Carla Schatz is involved in this, and she's the corresponding author, if she's involved in this, then it's probably good. Just like right off the bat, you can probably assume that. And I would be very surprised if it wasn't good. But it also shows kind of the structure of how this is done. And if I haven't explained this ad nauseum yet, the first author is the person who does the most work, right? Have I explained this to you guys? Not seeing any nods of any type at all. Speaker. Okay, thanks for all of your enthusiasm. So I'll just explain it anyway. First author, this is what you want as a young scientist. You want to be the first author. When you guys do your capstone, which really starts, the planning starts, I would say started now, sophomores, but through junior year, you're gonna pick something you're fascinated in, and you're gonna try to come up with a question, a project idea that hopefully will be something you can actually run experiments or collect real data on. And you might have teammates. You might say, okay, you know what, I'm going to work on this large, ambitious project with another, person. Who becomes first author is the person who does the most work, essentially. So there's a bit of competition there, and that's healthy competition. At places like Harvard, they're well known for the competition being like cutthroat, where people will undermine each other to try to get that first author position. But the most important positions on a paper are first author and last author. Last author is the person in whose lab it's done. So if we were to publish your capstone work, it would be, Lily Murphy, somebody else, and Chris Charleglio. I'd be last because it's my lab. Okay. They're also weird. Other institutions have weird. Lily, you were the first one I looked at. Sorry, I'm using you as the example. The other institutions that have weird rules. Sometimes they'll just do it alphabetically, which really hurts the first author, because then the person alphabetically is the first author, and everyone will just assume they did the most work. I find that really stupid. Other times it'll be simple rules like, oh no, it's just whoever makes the most figures, or whoever is going to stand to gain the most from it, or I don't know. We're gonna go and put Mr. Pompey on it because he's the principal. He hasn't done anything for it, but we'll put him on it. They have some institutions do that. It's kind of annoying. But now that you understand the structure, she's pretty old school. So if you go back to figure 1 for me. These, okay, so figure 1, if you're looking at this stuff here, this is a classic type of experiment. Actually, so is this thing over here. That's a classic experiment, a dot plot. But this one with all the colors that are really pretty is a type of method that right now we have scheduled for not this summer, but next summer, that I'm going to teach you how to do that. If you know how to do that, man, you are gold to get into a lab. Like you walk in, you tell the principal investigator, the PI, maybe it's Carla Schatz if you go to her school. Actually, I already know how to do immunohistochemistry or immunocytochemistry. And so what that involves is there's an antibody against a protein of interest. So what's the primary antibody for here? Who was doing this figure? Was it Andrew?
Yeah.
Andrew, what's the primary antibody? What are they actually looking at?
I'm assuming you're looking at little RB2, maybe.
Let's see. So she's also very good, very precise about writing. I just lost everybody. Am I frozen? Can you guys hear me?
We can hear you.
Yeah.
I just lost all of the video from everybody else. I don't know why. Okay. She's really good at writing figure legends. So for A, it says diagram. It's this. I'm talking about C though. So this is confocal images. That's a type of microscope that's very expensive, uses a laser. And see binding to LILRB family members in vitro. So these were transfected, MIC tagged, and immunostained with antibodies against C4D and MIC. Yeah, cool. So that's what the antibodies are against. So you take that, and then you use something called the secondary that has a fluorescent tag of a certain color. You hit it with light, and then it vibrates and then releases a photon. So freshmen, we'll talk about that briefly in chapter 7 of the textbook, which is the next thing. Oh, you guys have a test tomorrow. I forgot about that. I need to make that. Yeah, almost forgot. Luckily, somebody reminded me earlier today. Uh, so you can kill her later. Yeah, so the reason I'm stopping you to point that out is this is a really cool method that I do want to teach you guys. I think it's something that we can actually learn at Holy Cross. It can be tedious to do, but you get great results. Okay, I'll stop talking. But
All right, so we left off figure 1C. I said overlapping, more binding. Little RB2 had the most compared to all the other ones. So now figure 1D is basically just 1C but in numbers, right? So it's measuring the binding of C4D with three receptors being little B1, B2, and B3. So if you look here, B1, the lowest number right here, not very much correlation, not very strongly binded to C4D. B2, very close together, much higher on the scale. B3 also very high, but the dots are quite spread out, meaning that, C4D sure is going to be more receptive at like the very peak, but they're not, the dots are not close together and it doesn't show a good relationship between B3 and C4D. B2 seems to be the best based off 1D. Now in 1E, if we look at the shape of this graph, that's the most important part. It shoots up and then slowly starts to go up, but it seems to be going down too. It seems to be flattening out, right? So if we look here on the x-axis, it says C4D, how much C4D we have, right? In the very low numbers, like from 0 to 50, the graph goes up very high. In the bound C4D, which is the C4D connect actually, inside the receptor B2, um the bound C4D is quite low when you also have little C4D because again, receptors can only hold so much proteins. You can, if you have a receptor with 100 proteins, you put in 20, there's still 80 seats left for the proteins to go into. So you're going to have little bound C4D in this case. Now, this thing KD here, it means how like tightly attracted the cells are together in the receptor. That's what KD means. An important thing to note is that the balance for KD, which is like how do I say this? Is basically the balance for like molecules that are inside the receptor or proteins are inside the receptor and proteins that are, still swimming around and moving around, right? That's the balance, I'm pretty sure. So if you look here, they got 37
Balance.
from
What does that mean?
looking at the graph from the data they collected, and then finding out that they reach some sort of 50% equilibrium at around 37 nanometers. So what one is just saying is that, the balance for KD, the attraction between the molecules, was at 37. And a lower number means a higher attraction. So that must mean LRB2 and C4D have a very, very high attraction rate. Now, figure F, it is very similar to E, except PIRB, I'm pretty sure that's the receptor in mice, and they also found that the KD was 56, which is I'm pretty sure it's still a high number or like a high percentage of attraction between molecules. Okay. Now figure G and H, they use something called SPR. I don't know exactly what that is to test the binding in B2 and B1. So they found that B1 here, it was very low, right? There wasn't much signals received when we were doing this certain process, right? But in B2, when they ran C4D through this experiment and measured the actual reactions, it spikes up very fast and also plateaus and flattens down very fast compared to B1, which doesn't seem to have, any significant increase or decrease.
Is that it?
Can you
All right.
guys hear me?
Thank you. Yeah, we can.
Okay, I'm having technical difficulties. Had to restart this computer and then I just got kicked out of this one on my laptop. So sorry. Keep going.
Thank you, Andrew. Does anybody want to present figure one? Dylan?
Okay. Um, so figure 1I. So after they figured out that that LIR LILRB2 was like the best for binding, um, and like the PIRB, they decided to test they called it a wild type. That's just like a normal mouse, and compared to a mouse that doesn't have the PIRB receptor. They said PIRBKO, which is just like means it doesn't have the receptor. And so that first like two boxes. Do you mind if I draw on the slides?
That you could draw.
Okay. So something I wanted to point out was in the this is the wild type here. You can see like these like lines, and they're really similar to the lines of the TUJ, which is like this one here. And that just means that like the wild type, the mice that have the PIRB receptor are binding more to C4D, and that's pretty much what they wanted to test. And that's compared to the PIRB KO, the ones that didn't have the receptor, they're really not binding to the C4D. Like the C4D is not binding to the receptors. So that's really what they wanted to prove in that one. And then that third like picture, it's just like both of the two combined. And then they said DAPI, and that was just used to um like say where the nucleus of the neurons were. So like you can see like the blue spots on the top one and then purple on the bottom. That just shows where the nucleus is.
That is
And
beautiful.
lastly, to the far right of um I, they kind of just zoomed in on specific, just like parts of both like tests. And you can see that it's not really easy to see, especially on these slides, but the top, the normal mice, normal mouse, um, it has more of the C4D around the like axons of the neuron, and that just means that that C4D is binding to PIRB receptors. And then J is pretty much just saying what I just said. It's saying that the wild type is bonding more, like the C4D is bonding more to the wild type like receptors compared to the ones without the receptors. So, and then that third, the one down here, is that's just subtracting both of them and just the difference between them. And that's kind of about it for J. Uh, K I thought was kind of hard to follow, but what I really got out of it is that so they wanted to test so they figured out that LILRB2 and PIRB are the C4D is bonding to them. So they just wanted to see which domains it's bonding to. So it can't read it here, but they found that D1 and D2, the ones like you can see in red, are the ones that it binds to the most, and that's pretty much all it's showing. I was kind of confused on like on the bottom it says uh HLG and then like minus FC. I wasn't really sure what they were talking about on that bottom there, but anyways, um, sorry, do you have any idea?
Which one are we looking at?
For K at
K,
the
like
bottom.
where
I
it
don't know
says
what that
like
is
right
either.
here on the bottom, HLG dash FC or whatever that is.
So FC's flow cytometry usually, but let me see. Okay. Diagram, extra domain mutants. Uh, it's a, oh, yeah, I didn't understand that either. Here's, I'm going to shoot from the hip here because this is, if I'm right, it's important. If I want to show you how different mutations in a gene lead to changes in structure, then this is the type of drawing you'll do. D, in this case, would mean domain. I'm surprised she would use this nomenclature D1, D2, D3, D4, D5 because that nomenclature actually in neuroscience means dopamine receptor. But her I don't know if this is a grad student, Barbara Brott. I don't know if that's a grad student or postdoc. Maybe they thought that using D for domain would make sense, but domain is just a piece of a protein that has some functional meaning. So the first domain, the second domain, the third domain. And so if you look at the dashes that are happening like in the third column there, that would mean that it's removed in that particular mutant, I think. Yeah, mutants, yes. I think that's what it is. Does that
And
make
then
sense?
the
So
yeah, and then
how would they work? Because you change the structure. Sequence, you see, equals structure equals function. So they're actually showing that structural change. Yeah.
and then the red, is that just like what does that mean? Like is that what binds the most?
That was kind of why I was confused.
They didn't even like say anything in the legend or anything.
Right. So then let's go to the paper itself. That was K, right? So H K requires that extracellular IG domains. It's the binding domain. Right.
That's the red.
Well, because if you read it's on page 2 of the paper. If you look at it's on the right column of text, second section where it says C4D binding requires D1, D2 extracellular domains. It says that it's not flow cytometry, FC means something else. LALR has 6, 1K. Secreted soluble tag domain muons were incubated, and they found that C4D could bind to D1, D3, and to D1, D4, but they could not do to D3 or D4, D3 only or D4. So I wish this was a little clearer. Keep talking. I'm going to try to fix this on my side so it's a little clearer. Okay.
Right, so that bottom thing is just like a way for them to tag it and know like what it is. I think the next one, L, it's really closely related. It's just saying, um, like it's just talking about which domains it binds best to, and you can see again, it kind of says it like the D1 and it kind of like D2, it were like the most like binded the best, I think. I think like the thicker the line, like the better, or like I'm not sure.
Yeah. So remember, I was saying that Carla Schatz is old school. She does all of these really well-established methods. There are newer, sexier ways to look at some of these things, but she just goes for the classic ones that we know work. So this one, this L is called a Western blot, and a Western blot is, I mean, there's a history to it. Obviously, it's kind of actually funny. The first one they ever came up with, a guy named Southern came up with a blot to look at Southern blots, look at DNA. So things that bind to DNA. And then somebody was like, oh, I could do a similar thing to see what binds to a protein. We'll call it a Western blot. Now, the guy's name was Southern, but they thought it'd be funny to name this thing a Western blot. And then somebody's like, oh, you can do the same thing with RNA. And they called that a Northern blot. So a Western blot is, we scientists, by the way, have an interesting sense of humor. But the Western blot is protein-protein interactions. So if you see a big black spot, then that means that there is binding between the thing you're putting in and those things. And you'll see down at the bottom, they always put some sort of positive control, which tends to be a protein you will always find in cells. Guess what protein that is?
Was it the
Is
same
it
one from?
DNA?
No, a protein, a protein. What protein do you always find in cells, Giada? I'm calling on Giada. You know what it is. Actin, correct. So that's almost always what it is. And I'm looking to see. I don't really say what the, at least in the figure legend. So you should say what in the figure legend what you actually use as the control. That's a positive control because you know it'll bind. So I would have to go to the methods, transfections, neurons, KD analysis. It's not, I mean, it's important, but keep going. I don't want to slow us down.
Okay, so I mean I can see the thickest one is with D3, D4, D1, D4. So I think that's like where they had the best like outcomes. And then K, L, and M are really like really it's really similar. It's kind of just like a summary. So M shows that the D1, D4, so like D1, D4, D1, D3 had like the best outcomes for binding. As you can see, it's at one there, and then the other one the side. Compared to all the other ones. And in the results, they said, or like I don't know if it was results or what, but they said that E2 had like good outcome. But I don't really see. I don't see that on here. But and then PRRB, kind of the same thing.
Yeah, it's just the
D1,
mouse
D.
version.
Yeah, they look a little different though, and they tested different ones.
Mm.
This that one, first one is D1, D4 for the um LIL, and it's D1, D6 for the other one.
Who's sharing their screens? Marcello, right? Marcello, do me a
Yeah.
favor. Do me a favor. End the slideshow, then start it again because I went in and replaced the pictures with higher quality images that we can see easier.
Okay.
So you have to end the slideshow itself and
Yeah.
then reshare your screen once it starts up again.
Oh yeah, it looks better.
Oh yeah, way better. Okay, good.
Can you see my cursor on the screen?
Yes.
Oh, okay.
Yeah. Okay. So that's a little easier to see. What was the point though? This is because we have 15 minutes really if we want to end on time. What was the point of figure one
I
altogether?
think
The whole thing.
yeah, it was to test whether test what um C4D binds to, and then
Yep, that's
after
it.
they figured,
Yep,
that's
good.
and then
That's it. Yeah. Good.
Yeah.
All right, thank you, Dylan. Marcello, next slide.
Uh,
Okay, does anybody want to volunteer to present figure two? Oh, okay. Andrew.
Right.
No. Wait. See if anybody else wants to. We don't want to
Oh,
dominate.
Okay. Does anybody else want to present figure two?
I was gonna present E, F, and G, but I didn't look through the rest of A through D.
Okay, you can do that.
Well, I don't want to skip A through D, but if somebody else volunteers to do that, I'll do E, F, and G.
Does anybody want to do A through D?
Okay, really quickly. Ready? I'll do it. So
Okay.
V-glute is the transporter for glucose. And this is so glute, GLU capital T, is the glucose transporter, but the version of it is what you find on vesicles. And vesicles carry stuff between wherever they're produced, the proteins, right? And then down into the synapse and then are released. Okay, so they're just looking to see what things are co-localized. And this is immunohistochemistry specifically in a patient. This is a human sample. So somebody with Alzheimer's disease, what are we actually seeing? Is this a possible mechanism where LALRB2 is important or the C4D is important? Do you see it downregulated in patients versus somebody like if I went and took your brain and took a sample of it, you guys wouldn't have any of the problems. So for this first part, they're specifically looking at a type of neuron called a pyramidal neuron. They're cortical neurons that play a whole bunch of roles. They tend to be excitatory. They're really beautiful. If you look at in my office at school, right behind my desk, the very central piece of artwork that I have hanging there is I believe she meant it to be a pyramidal neuron. So they're cool. And so A is looking for co-localization in these samples. And let's see, this is a 60-year-old. This is from temporal lobe. They're looking for pre and postsynaptic markers. So V-glute, since it's vesicular, you should only be able to see it in a presynaptic neuron before a signal is sent. And in a postsynaptic neuron, you would see the PSD stands for postsynaptic density. So this is pretty smart, right? If I want to know whether two proteins are in a synapse, I'm going to take something that marks the presynaptic neuron and something that marks the postsynaptic neuron so that they glow, and then co-localize it with a third thing to see if it shows up for any of these proteins, and does it. So if you're seeing them co-localize, which you are in box 1, then you can see that the LALR-B2 isn't there. C4 might be. See how it's white up at the top? Can you guys see my? Oh, you can't see my cursor. Hold on. You see right here. Okay, I'll clear that again. But that's kind of white. So it might be there. And then it seems to really be there in the C4D. Okay, telling you that that's actually there. And then if they look at specific synapses, they just zoomed in B. Okay, looking at synapse 1, 2, 3, and 4. And then they're trying to correlate and see whether these things go together. So V-GLUT and PSD95 should go together. C4D and PSD95 is higher, so that's going together. But you see it drop with the other ones. Let's see, D. I told you that these pyramidal neurons tend to be excitatory. In an older person suffering from Alzheimer's disease, are they still functional? So like percent of excitatory synapses, you can mark it for. So why V-GLUT? Why would you choose that as the marker for a presynaptic neuron? Because glutamate is the excitatory neurotransmitter. So you're marking an excitatory synapse. So if it shows up, you know it's an excitatory synapse, and you're trying to see, trying to co-localize it with these other proteins to see if there's any effect. And let's see, the last one has 3 different patients, and it's showing how much of that C4D they have. But you take over, Cadence, and tell us what that does. Because the first ones are just setting up for the data, which is what Cadence is going to talk about.
So I hate these graphs or plots or
Why?
whatever because
Why?
I don't know, maybe I'm used to like simpler graphs or plots, but it was just like kind of
This is an actual gel. I mean, it's not actually gel; it's a piece of paper, but it had a gel stuck to it. Remember we ran gels over the summer, and you had the bands.
yeah.
That's what this is, kind of.
If I'm wrong,
So
the black stuff is proteins going to the gel, right?
yeah,
Okay.
the dark, so like where it's dark on the graph is
Yeah,
where the proteins are.
right. So the really, really super ridiculously dark stuff in F, what is that?
Isn't that where like the proteins are? Like,
I think that's what you don't like about it, right? It's here. Ooh, just took my ability to draw. This stuff here, right? Do you guys see that?
I
Um,
see it.
right. Is that what bothers you about it? It's so messy.
yeah.
That's the control. So
Okay.
the thing that matters is actually these bands up here. Okay.
Okay, that makes sense.
Mhm.
Okay, so figure E, that's where the synapses were isolated. Um, so we can see that I don't know if it's LIRB2 or LILRB2, and then C4D are detected in the synaptosomes, and then the PSD95, it kind of like confirms the samples that they contain the synapses. Um, and they use the bottom row, the TUI J1 is used as the control. It's just like a marker protein. Um, yeah. And then figure F,
So
um,
why though? Why would they do that? No, no, it's actually kind of think about it. Why would you let's start with the control. Why would you run a control?
because that would be like the base that you go off of, like the standard kind of,
And you would expect it to be there, right? There's a positive control.
right?
If it didn't show up, what would that tell you?
Like
The,
something's wrong.
yes, that you did it wrong. That's all.
Yeah.
That's it, right? Now, PSD stands for postsynaptic density. So why would you care about that?
I mean, if it's post, it's like what it is after. So like you
After the
kind of need
synapse.
to see the difference.
It's not about timing. It's about an actual protein in the synapse. If you have less of it, that means you have fewer synapses. Why would you have fewer synapses?
Alzheimer's?
Yeah, you see?
Yeah. And then figure F, this is where LIRB2 is pulled out of the synapse samples using an antibody. Um, LIRB2 is present in the human synapses. Um, it's detected in both the control and the Alzheimer's brain.
Mhm.
Um,
You see a difference?
well, I mean, it's darker.
Yes. What does darker mean? So this is the control.
More of the protein is when it's darker.
Exactly. Here's the Alzheimer's brain, is the second one I'm drawing. What are you seeing?
It's like dissipating, there's less of it as it
There's
goes.
less of it there. If there's less, so what they're saying is it's kind of correlation is not causation. But the authors are doing a really good job of showing all the circumstantial evidence that they seem to go together. Like if LILR or Liller B2 is present, then you get up here lots and lots of synapses. If it's here in this middle sample, which is Alzheimer's disease, notice it's lower, and notice the PSD is also lower, right? So that means, and they can do this, they can manipulate it in a mouse. If I knock down how much Lil or B2 I have, then I'm also going to have fewer synapses. So maybe if I protect it, right? Is that, I'm getting ahead probably, Marcello and Christopher, right?
Yeah.
Yeah. Okay. You're doing a good job, Cadence. So keep going.
I'm trying. I was kind of
Mhm.
confused because like I didn't really get a chance to read like as thoroughly like the paper, but um
But you can see that, right? You can see the differences.
yeah.
Okay.
Um figure G, this is the whole brain life states are compared to the synaptosome fractions where you could see PSD 95 has like a lot of the synapses. Um which like goes back to F where like it's really like happening, like they're very present in the human brains. So like it's kind of used to like support everything that was like in F.
Mhm. Yeah, good job.
Yeah. Thank you, Cadence.
This was it trying to like, it's just trying to prove that the C4D is what's causing the Alzheimer's.
Hmm.
Uh,
Causing, not just there by happenstance, but is actually one of the things that seems to be a major
yeah.
motivator for getting Alzheimer's. Yeah.
Uh, figure four. Does anyone want to do figure four? Do we have time?
Yeah.
Uh,
I can. I don't know how to raise my hand on here. I think it's
That's fine, do it.
okay. Um, so A, if I read the figure, I can't tell from like just the picture, but in the legend, or yeah, it says that it was when they gave it to the mice for 4 days, the C4D in the visual cortex for all 4 days.
Mhm.
And then B, you can see the C4D in the DAPI. So that just, I'm pretty sure like the brightness on the C4D shows like it reaching the tissue that it was trying to get into the mice. And then C, is that,
Just a neuron.
oh, that's just a neuron? Okay.
Just a neuron. Yep.
That's just showing like what it looks like inside.
Yep.
Okay. And then 4, is that what the stuff inside the neuron like looks after the treatment?
So the box in C that's drawn there
Yeah, that's like it
is,
broken down more.
yes, it's a
Okay.
zoom in.
Yeah.
Yeah.
Do you know what those are called?
What's called?
Those like the lines. The
In
lines
D?
say like yeah.
Oh, yeah, those are axons. So, or actually, no, these might be presynaptic. D, D, basolateral. Oh, the dendrites. They're the receptive parts, which means that you are looking at a postsynaptic cell that receives a signal. The
Okay.
stuff that had the PSD. Yeah.
Go ahead, Cadence.
This isn't like a question like just for you, like if anybody knows the answer, but you said that they tested like the mice's like their visual like part like visual cortex, is that what you said? But like
No, the mice were given the C4D through the visual cortex. That's what it said in the bottom.
The reason for that is just it's easier that way because their visual cortex isn't like ours. So our visual cortex back here, and their visual cortex is actually kind of on the top. So you just make an incision and remove their skull, part of their top of their skull. It's a craniotomy. And then you put in a cannula and then cover it with cement to hold the cannula and place it. And that way it's just easy to get to. So they just chose that.
okay.
Yeah. Because they also have pyramidal cells. It's a cortical cell. It's just a plain old cortical cell. Yeah. But what are we seeing? Like, what is there a difference?
Um, yeah, I think so.
Tell us what it is. Because that's E, right?
Yeah, E,
Mhm.
I think so it says that the syn uh what does that say? SPINE. The
Fine.
the spine density.
Yep.
So it shows, I'm pretty sure that shows that the C4D lowers the synapse numbers in the normal mice, right?
The C4D lowers? No. No, doesn't lower
Is it higher?
it. Does it lower it?
It says in the talk about like in white mice, it like decreases spine density compared like decreases a significant amount compared to control.
Yeah.
It de
Yeah, so lower.
Figure E is the same thing as figure D, just like in a different way. Like if you look at the like their labels, they're like the same thing. So like if you're like having trouble with D, just look at E because they're the same thing.
Yeah.
yes. Yes.
So it's lowering the synapse numbers in the normal mice.
Right. So why isn't it lowering the synapse numbers in the knockout? Right. That's what NS means. Is right here.
Yeah. That
What? Why
line.
isn't it? Why isn't my pen working? And why isn't it significant? Not significant. Right here.
Why is what not?
Why is it that when you infuse C4D into a knockout mouse who does not have this gene here,
Mhm.
that C4D does not cause spine loss? Why?
because it needs PIRB to cause spine loss.
Yep. So go to the figure 5.
Boom. Okay.
Does anybody want to present figure 5? No?
No, you should.
I should?
Yeah.
Okay. So this is showing, so figure A is a simplified drawing of like a healthy excitatory synapse. And um the I don't really know how to explain this one. Um the I don't know how to explain it.
Well, so in A,
Yeah.
LILR-B2 is the PIBR that you saw on the previous PIRB that you see on the previous slide, right? They're the same
Yeah.
thing. It's just the human version of it. And what does it do? It's a receptor. Well, what's the thing that activates the receptor? That was the question. And so in
C4D.
B, it's C4D. So when you have C4D present, when you release C4D and it binds to LILR B2, it causes the synapse to decay, to degrade. But if you don't have the receptor, which is what you see in figure 4 in the knockout, if you don't have the receptor, then C4D can't cause damage. So that's it. What are the implications of that for us poor people who actually carry the gene, like me?
Wait, if they don't have like the PRB thing, how would they
Yeah,
get rid of synapses?
they can't really, not as efficiently. They're usually redundant mechanisms, but you're not going to be able to as easily.
Uh,
Yeah. And why is it important to get rid of synapses in the first place?
Because some synapses when they get like trashed, you have to get rid of them like and replace them. I don't know explain it. Like
That's one reason. Yeah.
Mr. Cream was like kind of explaining it to me earlier.
Yeah.
I think it was like when you have more synapses, the weaker ones, like they kind of take down the stronger ones. So if you
Yeah.
remove the weak ones, the strong ones get even stronger.
Yeah. And that's called Hebbian plasticity, where neurons that fire together wire together. So they actually have to be used in order to be strengthened. And actually, Carla Schatz is the one who kind of came up with that. Heb's the one who showed it, but she's the one who came up with the saying, neurons that fire together wire together. Um, and so in this case, it would be bad for people or animals to not have LILRB2 because then they can't really prune their synapses as efficiently. However, later on in life, you kind of don't want to prune as many synapses as Alzheimer's disease patients do. Cadence.
This might be a stupid question, but like, you know how like in proteins, like when you take a drug, there's like the competitive inhibitors and it like blocks like the interaction from happening. Couldn't like later in life, if you wanted to like maybe not prevent Alzheimer's altogether, but like you could invent like a drug that can prevent the synapses from happening?
That's not stupid. That's the point.
That's what I was just gonna say. You could create something to block the receptor so that C4 can't bind to it and shut down the synapse.
Mm-hmm. Okay. Dylan?
I just had a question about Alzheimer's. Like, when you have it, is it just one specific part of your brain? Like, can it be like
It's cortical.
specific?
It's not subcortical, it's cortical, but there's a weird phenomenon that happens in Alzheimer's brains where for some reason, we don't really understand why, but for some reason, motor cortex is spared. So we all know people who had Alzheimer's. We probably are related to them. My grandmother died of Alzheimer's, but till the day she died, she could have kicked my butt in basketball. She was very, very coordinated. She still had her motor skills. That's very different than ALS, where you lose your motor neurons, but not your cognitive neurons. It's like almost the opposite.
So it's not like you can like target that one specific part of the brain that has Alzheimer's. Like
No, it's
you would
pretty
have to
widespread. Yeah, pretty widespread. But what Caden said is kind of the point here, right? And let me take that further. My kids who are 2 years old have about 2,500 synapses per neuron. I have about 2,500 synapses per neuron. You guys have 10,000 synapses per neuron. Why? Why? Is something happening in you guys that doesn't happen all the time?
Is it puberty?
It is puberty, yeah. So you're having an uptick in hormones that are adjusting your metabolism and actually causing rewiring of the brain. And there's a bunch of other things that are involved in this. For example, selecting which ones to prune during puberty, or that's a serotonin-based thing. If you have serotonin at the right time in the right place, you'll actually prune it. But if you go and you screw that system up, so I've talked to you about this a little bit, so I think this is interesting. So if you need to leave, please do, but I think you'll find this interesting. Antidepressant use in adolescents like you guys is very, very dangerous. What we found in, I think 2010 is when we really, really discovered this, that people your age who are using antidepressants are actually increasing your suicidality. And it's supposed to be an antidepressant. It's not a thing that causes suicide. Why is it doing that? And we think it's because the pruning that's happening of synapses that don't matter, that pruning's happening under serotonergic control. But if you are taking an antidepressant, it's completely screwing up your serotonin system. So you have way more of it, which means it doesn't know what to prune correctly. So like, why are kids generally more creative? They have crazy ideas, right? They're generally more creative than adults because those kind of useless synapses get pruned. They're supposed to be pruned. You know what's not supposed to be pruned are the synapses of your life, the things you're supposed to remember. So Alzheimer's does that. You see? But it's really interesting. There are certain disorders where the synapses are the synaptic structure is wrong. Like what? Can you name a disorder where it's a developmental disorder where synapses are malformed or misnumbered? Wild guess. If I'm asking you, it's something you know.
Down syndrome.
Down syndrome.
Down syndrome a little bit, yeah. Not what I was intending.
Is it
Autism?
Autism, yeah. So in autism, one of the reasons why in autism people who have autism get overwhelmed by stimuli is they're actually not pruning correctly. Sorry, no. They have too many synapses. Too many synapses is bad. So what's happening is instead of being able to focus on a single stimulus, everything else that's coming into their brain their brain is treating it all as if it's important, salient stimuli. So they quickly get overwhelmed. Well, what's cool about this for people who have kind of like low-level autisms is like, so it's not super severe. When they go through puberty, puberty will allow this process to occur where you're pruning away synapses that don't matter. And so on the other side of puberty, when you hit adulthood, your brain is more normally organized than before. And I'm not saying that the autism disappears, but in a lot of people, it improves greatly. Cool. What else you got for us? Favorite new word?
Leaved.
And does it belong in the proceedings of the National Academy?
You said yes.
Yeah, this is a good journal. I was an editor for this journal for a bit. So, and you have any other slides?
No, that's it.
Just like sources.
Cool. Okay. Well, any questions? Comments, concerns?
Why did you have to use the mouse brain instead of any other brain?
Mice are our model of choice for how it's very simple. You can store 5 of them in a shoebox for long periods of time. They love it. They're just one of the models of choice. I'm trying to get us mice at school.
Ooh, don't do that. Don't do
Why?
that. Mice?
Yeah,
What if they
Ooh.
like escape and like run around the school?
we got to make sure they don't, because that would be very bad because they're not actual wild type mice. They're not natural. They're usually the ones I would be bringing in would have mutations so that we could study specific things.
Is there like any other like diseases that happen in the brain that like would like if they came up with something to prevent the synapses, like it could also like improve the effects on like other diseases or like just Alzheimer's?
The dirty secret about Alzheimer's research is that Alzheimer's is kind of a catch-all. Like it has certain characteristics, but it doesn't always behave the same way. So if you're God, you would actually be able to say, yeah, there's Alzheimer's A, Alzheimer's B, Alzheimer's C. They're different versions that play out differently. It would help with all of those, but it would also help with like when you have Parkinson's disease, you all know what Parkinson's is. Just how does Parkinson's kill you? Have you ever thought about that? How do you die from Parkinson's? Anybody know?
Your brain stops working?
Yeah, that's pretty good. That's a pretty good answer because it's not just a movement disorder where you have these hill rolling tremors. It's not just movements. That's the annoying part. But actually, when you have those type of tremors, we know that you've actually already started having serious brain damage. So, but the scary thing was that if you're at the point where you're actually having these symptoms, it means you've already started having the mental symptoms where you are having hallucinations, you're not able to make decisions, you're forgetting things because that's the thing that ends up killing you. Your brain kind of shuts down and you have a bunch of synaptic problems that look just like Alzheimer's. In addition, you have these little things stuck in your brain called Lewy bodies, these little clusters of protein that are just killing cells. So that would be one of them. Probably ALS, probably Charcot-Marie-Tooth. There's a bunch of disorders that would apply to some sort of cure for this, some sort of treatment. Okay, good. Was this a better paper than last time? Yeah, scientifically, yes. Maybe
Yeah.
not as fun. Maybe not as fun because we couldn't destroy it. But it was. She's a great scientist. So, all right. All right, we're going into overtime. See you guys tomorrow.